today's session we will be trying to understand the internal structure of kidney, that is vertical section or longitudinal section of kidney. In the earlier session I had told you about the morphological structure. Now let's have a look at the vertical section. This is the vertical section wherein the outermost covering, which we had earlier discussed as the renal capsule. This is the renal capsule. That's a fibrous protective covering. Now rest of it, the internal structure, can be actually divided into the peripheral cortex and the central or the internal medulla. So this is the renal cortex and rest all is renal medulla. So renal capsule would be enclosing renal cortex and renal medulla. See, the blood vessels, the artery and the vein, all their branches would be distributed majorly in the renal cortex. The branches of renal artery and the tributaries of renal vein, they would all be scattered majorly in the renal cortex, and therefore, as I have told you earlier, the kidney, it has reddish brown color. That's because of the blood vessels which are distributed in this layer, which is just next to the outermost covering capsule. So that is the outermost layer. Then the medulla, peripheral part of medulla, the part which is facing the cortex, it comprises these triangular units called renal pyramids. You can see these renal pyramids. They would be 6 to 13 in number in every kidney. Each pyramid would have its broader end called the base facing the cortex and narrow end called apex facing towards the center of the kidney. That's here. Now every two adjacent pyramids are separated by a minor gap which is called as the column of Bertini. Column of Bertini. So every two pyramids will be separated by column of Bertini. Now next to the pyramids would be the central space or the cavity. Now as could be seen, the cavity would be formed by joining of small pocket-like units present near the apex of each pyramid. These pocket-like units are known as the minor calyces. Minor calyx would be singular. So each pyramid have a minor calyx at its apex, and number of these minor calyces then join and form the major calyx. So this central cavity would be the major calyx, which then continues into relatively shallower cavity called the renal pelvis, that then runs into the ureter. Okay, so the outermost covering would be renal cortex, further covered over by a fibrous renal capsule. Cortex would be the layer wherein most of the blood vessels are distributed. Then there would be these renal pyramids, 6 to 13 in number, every two adjacent pyramids separated by column of Bertini. Next would be these tiny pocket-like formations called the minor calyces, which then join into the major calyx. That continues into renal pelvis and then it opens into the ureter. Now, as could be seen, each pyramid has a fiber-like appearance, fibrous appearance. That's because of the functional units which are contained in it. These functional units are called as the nephrons. See, the pyramids and the cortex, they are not separated by any membrane or cell. Rather, the two layers are not so distinct from one another. That is because the units meant for filtration of blood, they are partially enclosed in the pyramids and partially in the cortex. So the boundary between pyramids and cortex is not a clear boundary. These two layers appear to be almost intermerged. Contained majorly in the pyramids are one part of the functional units called as a nephron or uriniferous tubule. and rest of it is contained in the cortex. Now, what is this nephron, or what is the uriniferous tubule? Let's get to understand that. This is entirely the uriniferous tubule or the nephron, which would consist of this cup-shaped structure called as the Bowman's capsule. Next to that is a narrow part, that's a neck. Then this entire coiled tubular structure is called as a renal tubule, which would include the first coiled part, that is the proximal convoluted tubule. Then this relatively narrow U-shaped tube, that is the loop of Henle or Henle's loop, followed by distal convoluted tubule, which then joins it to a collecting duct. Now, when we talk about just Bowman's capsule and renal tubule, this is the nephron. And when you include the collecting duct with it, it becomes the uriniferous tubule. See, each kidney contains about 1 to 1.3 millions of these nephrons. But though the number of nephrons is 1 to 1.3 millions, the number of collecting tubules, if you include along with it, the number goes relatively lesser. Because as could be seen, a number of nephrons would be joining into the same collecting tubule. Hence, this whole thing would be the uriniferous tubule. When you exclude the collecting duct, it becomes the nephron, which would further comprise the Bowman's capsule, the neck region, the proximal convoluted tubule, the Henle's loop, the distal convoluted tubule joining into the collecting duct. That is what a nephron or a uriniferous tubule will be. Out of this U-shaped part is mainly covered inside the or enclosed inside the pyramids, so renal pyramids, and rest all, that is most of the proximal convoluted tubule, most of the distal convoluted tubule, they would be contained in the cortex. And that's why I said the pyramids and cortex are not so sharply separated from one another, because part of this unit is contained in the pyramids, part of the unit is extended into the cortex. The collecting tubules, they would be majorly contained in the peripheral part of the pyramids. Now, that's the overview of a nephron or a uriniferous tubule. The Bowman's capsule, see here whatever you are seeing is in a sectional view, longitudinal section. Otherwise this would look just like a cup-like thing. This cup-like structure, the Bowman's capsule, you can see there are two layers. This is the inner layer, this is the outer layer. Both the layers are made up of squamous epithelium. Squamous epithelium, flat polygonal cells placed edge to edge. But the inner layer is called as visceral layer, outer layer is called as parietal layer. Inner layer or the visceral layer consists of a squamous layer from which a few cells are modified into podocytes, that is tiny cells with microscopic extensions, podocytes. So some of the cells in the inner covering of Bowman's capsule, they are modified into podocytes. What is their function? That we will get to know later on when we talk about physiology of urine formation. Between visceral layer and parietal layer, there is a cavity which is then called as the Bowman's space or Bowman's cavity. Next is the neck region. Now, neck is made up of ciliated cells, ciliated epithelium. It's a narrow part of the nephron. It's lined by ciliated epithelium. There it is, proximal convoluted tubule. Proximal convoluted tubule and for that matter distal convoluted tubule, they are both lined by cuboidal epithelium. But proximal convoluted tubule would be lined by cuboidal epithelium which has microvilli in it. You must be knowing that as brush border epithelium. So this part of nephron, the proximal convoluted tubule, is lined by cuboidal cells with microvilli. It's an absorptive modification. Distal convoluted tubule, on the other hand, would be just plain cuboidal epithelial cells. The Henle's loop, relatively a narrow part of the nephron, it is lined by squamous epithelium again. So Bowman's capsule and Henle's loop are lined by squamous epithelium. Proximal and distal convoluted tubules are lined by cuboidal epithelium, one with microvilli, one without microvilli, and then it joins into collecting duct. Collecting duct which continues further. Let us relate it to the blood flow. Now we can see the nephron as well as the blood vessels. See, this is the renal arteriole, which gives out a branch towards the cortex basically. This is the renal vein, which would collect the smaller venules and would be joining into a renal vein. This is the collecting duct. This is the Bowman's capsule, proximal convoluted tubule, Henle's loop, and distal convoluted tubule. That is the general structure. Now, as could be seen, renal artery sends in a branch towards each Bowman's capsule. So, if I say that there are about 1.3 millions of nephrons in the kidney, renal artery would be giving out 1.3 millions of branches into it. So every Bowman's capsule receives a branch of renal artery. This incoming vessel or incoming branch of artery or arteriole would then be called as an afferent vessel or afferent arteriole, which then divides into numerous branches or capillaries. This bunch of capillaries is then called as the glomerulus. These glomerular capillaries rejoin and form the efferent vessel that then continues further. So this is the glomerulus that joins into another vessel, another arteriole, that is called as an efferent or outgoing arteriole, which then runs down towards the neck and starts dividing once again into capillaries. Now, one thing we must understand, the incoming vessel is an arterial vessel because it contains oxygenated blood. Outgoing vessel is also an arterial because now also it contains oxygenated blood. And then it starts dividing into capillaries. This bunch of capillaries we have called as glomerulus, while these capillaries, they are named as peritubular capillaries because they are in the periphery of the renal tubule, or they are also named as vasa recta or peritubular capillaries. You can see these capillaries, they cover the entire nephron. Complete nephron is surrounded by these capillaries. There's a very rich, very dense network of these capillaries around the nephron. Then the capillaries joined together form a venule that then continues into renal vein. Now here the capillaries are becoming venous capillaries. That's because during this passage of blood, the oxygen is being utilized for some purpose. So the blood becomes deoxygenated, which then is collected by the capillaries that join into the venules, and then it continues into the renal vein. That is how the blood flow can be explained. Now let us understand the artery and its branches are mostly in the renal cortex. Almost the same about vein and the tributaries. They are in the cortex, but the Henle's loop and the capillaries around Henle's loop would be contained in the pyramids. That is how the distribution can be explained. Keep watching these videos, like them, subscribe to them, and share amongst your friends and family.